Hello students, today we will be taking up metabolism of carbohydrates. So the contents would be, we will have a brief introduction about metabolism. We will talk about various pathways of carbohydrate metabolism. We will talk about in this video about glycolysis, various steps involved in the glycolysis, then energetics or the balance sheet involved in glycolysis, finally regulation of glycolysis and clinical importance. Now, in our previous videos, we talked about carbohydrates. In fact, chemistry of carbohydrates. We gave a definition of carbohydrates. What carbohydrates are? We said carbohydrates are polyhydroxyaldehydes or ketones or their derivatives. They are known as hydrates of carbon. Secondly, we also discussed the classification of carbohydrates. So, in order to revise you people, carbohydrates are classified into three prominent classes, that is monosaccharides, we have oligosaccharides and polysaccharides. This classification is on the basis of pattern of hydrolysis. Then we also talked about structure of glucose. Structure of glucose can be open chain structure. This is the open chain structure of glucose. As you can see, this is open chain structure of glucose or straight chain structure of glucose. Then we have the ring structure of glucose. This is in fact ring structure of glucose. This is Haworth formula of glucose. This is ring structure of glucose. Then we need to know what is alpha D glucose and beta D glucose. As you see in this very ring, this is Haworth ring, a representation of glucose, the ring structure of glucose. You see this carbon atom number 1, H lies above the plane of the ring and OH lies below the plane of the ring. So this carbon atom number 1 is an asymmetric carbon atom. It creates two isomers known as alpha isomer and other one is beta isomer. So this is now alpha D glucose. Now glucopyranose means that this particular ring is a pyrene form of heterocyclic compound. Pyrene form means heterocyclic compound with oxygen as heteroatom. Similarly here also, if OH group is present above the plane of the ring and H below, then we will call it beta D glucose. So alpha D glucose and beta D glucose are anomers. So anomers are that type of isomers which differ in configuration at asymmetric carbon atom 1. This is also known as anomeric. This is the brief revision pertaining to metabolism of carbon which you should know, and we have already discussed in our previous videos. Kindly go through those. Now, what is metabolism? This is our current topic. Metabolism simply means assembly of biochemical reactions. It is not a single reaction, assembly of biochemical reaction, or you can say enzymatic reactions, because all biochemical reactions within the body are carried out by enzymes employed by organisms for what purpose? For the synthesis of certain cell materials, utilization of energy from their environment. This could be a precise definition of metabolism. And secondly, metabolism involves two important processes, catabolism and anabolism. In fact, we can also say metabolism means sum total of catabolism and anabolism. Now the purpose of metabolism is, or the function of metabolism would be to get chemical energy from what? From breakdown of energy rich nutrients. For example, glucose is energy rich compound, lipids are energy rich compounds. So when they are subject to breakdown, we get energy, chemical energy in the form of ATP, adenosine triphosphate. Now we have to convert these nutrient molecules into precursor of cell macromolecules, because unless and until we subject proteins to degradation or carbohydrates to degradation, then only important cell molecules like DNA etc. would be generated. Later on, we have to assemble precursors of these very macromolecules like proteins, lipid, carbohydrates in the formation of important cell molecules, for example nucleic acid. Nucleic acids contain proteins, nucleic acid contain nitrogenous bases, adenine, guanines etc. Now catabolism means breakdown of macromolecules into simpler products with utilization of energy. Energy means in the form of ATP, adenosine triphosphate. Whenever we talk about energy in biochemistry, that means we are talking about ATP. So catabolism means degradative pathway. Catabolism means breakdown of macromolecule, larger molecules into simpler ones with utilization of energy in the form of ATP. Now, anabolism means it is a synthetic pathway in which smaller molecules are assembled and energy is utilized here. Here, energy would be generated. Here, energy is utilized. So, this is catabolism and anabolism. Now, what are the metabolic pathways of carbohydrate metabolism? How carbohydrate metabolism takes place? It takes place by 6 pathways. First of all, we have glycolysis. This is the major pathway of carbohydrate metabolism. Then we have TCA cycle or Krebs cycle, also known as tricarboxylic acid cycle. Next, we have gluconeogenesis. Then we have hexose monophosphate shunt pathway. HMP shunt pathway. Next we have glycogenolysis, and lastly we have glycogenesis. These are the 6 pathways of carbohydrate metabolism. Some of these pathways are catabolic pathways, some of these pathways are anabolic pathways that we will talk about later. So here we are interested to talk about glycolysis. Glycolysis means breakdown. So it is a catabolic pathway. Secondly, we need to know where does glycolysis take place. That is site of glycolysis. That is extra mitochondrial part of the cell, that is in cytoplasm. Now glycolysis is encountered in 10 step. It is a 10-step enzyme catalyzed reaction. It can be of two types. It can be aerobic pathway or anaerobic pathway. That means it can take place under aerobic conditions, mean in presence of oxygen. Anaerobic means in the absence of oxygen. Then you find in glycolysis various reactions. Some of these reactions are irreversible, some are reversible. So out of 10 reactions, we find 3 reactions are irreversible. First reaction, third reaction, and the last one, they are irreversible. Rest of the reactions are reversible. So you have 7 reversible reactions and 3 irreversible reactions. It is carried out in 2 phases. We have phase 1 and phase 2. Now phase first is supposed to be priming phase. Phase first is supposed to be collection phase, in which various carbohydrates, glucose, monosaccharide, fructose, each and every carbohydrate is assembled. It is prepared to undergo glycolysis. Now phase second is the actual catabolic or oxidative phase. Now metabolism, we can say in briefly, it involves catabolic reactions, it involves anabolic reactions, there has to be some balance sheet, byproducts are generated, it is an organized pathway, it yields energy, D-glucose is involved, their enzymes are involved, there could be sometimes deficiency of these enzymes. Certain steps are very important, they are known as rate limiting steps. They could be carried out under aerobic or anaerobic conditions. Now we can see from this small figure that in metabolic pathway you have a precursor, then you get certain intermediates, and finally you get products. So this is known as linear pathway of metabolism. So glycolysis is a linear pathway of enzyme catalyzed metabolic reactions, where you have a starting material, that is precursor, then you get different intermediates, and finally you get products. Now what we find is we start with glucose, obviously alpha D glucose. We have to start with alpha D glucose. Now we have made you understand what we mean by alpha D glucose. Now glucose is converted into pyruvate. Now glucose is a 6 carbon sugar. It is a 6 carbon sugar, C6. It is a hexose. It is aldohexose. It gets converted to 2 molecules of pyruvate. It is a 3 carbon compound. Pyruvate is converted to 2-acetylcholine molecule, coenzyme A molecule. Then acetyl coenzyme is finally converted to carbon dioxide and water. Now pyruvate can meet different fates. It can get converted to lactate in muscles, etc. Or pyruvate can undergo alcoholic fermentation under anaerobic conditions, that is you get ethanol and carbon dioxide. So in glycolysis, glucose is subject to conversion to pyruvate. Now you see this is a D-glucose, this is straight chain structure of glucose, this is Fischer projection form of glucose, this is what we say cyclic structure of glucose, you can see, or the ring structure of glucose. Here you can get further clarification. Molecular formula of glucose C6H12O6. These are 6 carbon atoms, 1, 2, 3, 4, 5, 6. Then we see in glycolysis different reactions are carried out. There are 10 reactions as we said. Reaction number 1 is phosphorylation reaction of alpha D glucose. This is the enzyme involved for the conversion of glucose to alpha glucose 6 phosphate. That means we have a glucose molecule, to this is attached phosphate group under the influence of enzyme hexokinase, and phosphate group is obtained from ATP is also involved, magnesium ion is also involved, so that is phosphorylation reaction. We have to remember one thing, wherever you come to know about phosphorylation, the enzyme would always be a kinase, and secondly the ATP would be utilized in the reaction. ATP is utilized whenever we have a phosphorylation reaction. ATP is involved, kinase is involved, and secondly we have a metal ion, that is magnesium. It is used as a cofactor. Without this cofactor this reaction is not possible. Next reaction is isomerization. Glucose 6 phosphate is converted to fructose 6 phosphate. Enzyme would be phosphoglucoisomerase. Next reaction again it is phosphorylation of fructose 6 phosphate, which is generated here. Under the influence of, again we get a kinase enzyme, phosphofructokinase. You get fructose 1,6 diphosphate. Now fructose has been again, 6 phosphate has been against phosphorylated at which position? 1 position. Now you get 1,6 diphosphate. Initially it was 6 phosphate, now we get 1,6 diphosphate. Now fructose 1,6 diphosphate breaks. The enzyme is aldolase. This type of reaction is cleavage reaction. We get two compounds, dihydroxyacetone phosphate and D-glyceraldehyde 3-phosphate. This is the 3 carbon compound here, and another one, glyceraldehyde 3-phosphate. This is also a 3 carbon compound. Now you can understand, we started with glucose, it was a 6 carbon compound. We got fructose 6 phosphate, again C6 6 carbon compound. Then we got fructose 1,6 diphosphate, again C6 6 carbon compound. Now this 6 carbon compound was broken into 3 carbon compounds, that is dihydroxyacetone phosphate and D-glyceraldehyde 3-phosphate, 3 carbon compound. Then dihydroxyacetone phosphate compound again undergoes isomerization, under the influence of which enzyme? Triose phosphate isomerase. We get D-glyceraldehyde 3-phosphate. In fact, two molecules of D-glyceryl 3-phosphate. Then seventh reaction is transfer of phosphate from 1,3-diphosphoglycerate to ADP under the enzyme phosphoglycerate kinase. Reaction 8 is again isomerization of 3-phosphoglycerate to 2-phosphoglycerate under the influence of enzyme phosphoglycerate mutase. Then 2-phosphoglycerate, which we got here, undergoes dehydration by which enzyme? Enolase enzyme. You get phosphoenol pyruvate. This last reaction, transfer of phosphate from phosphoenol pyruvate ADP, here you get actually pyruvate. Now this very last reaction is a irreversible pyruvate. Here you get pyruvate molecule. This last reaction is irreversible reaction. So reaction number 1, as I said earlier, reaction number 1, reaction number 3, and reaction number last, this one, reaction number 10, they are irreversible reactions. Rest of the reactions are reversible. Now you can see from this chart now, glucose, we started with glucose, enzyme was hexokinase, we got glucose 6-phosphate, ATP is used as energy fuel, ATP is converted to ADP, magnesium ion acts as cofactor, you get glucose 6-phosphate. Now you have the isomerase enzyme, glucose 6-phosphate gets converted to fructose 6-phosphate. This is catalyzed by phosphofructokinase enzyme, you get fructose 1,6 phosphate. This is broken down by aldolase. You get dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. Glyceraldehyde 3 phosphate, this now dihydroxyacetone phosphate gets converted to glyceraldehyde 3 phosphate by this enzyme phosphotriose isomerase, and finally you get two molecules of glyceraldehyde 3 phosphate. Now glycerol diphosphate is converted to 1,3-diphosphoglycerate under the enzyme dehydrogenase. This is the oxidation reaction. This is the first oxidation reaction of phase second. This is phase second reaction. Remember this. Conversion of glyceraldehyde 3 phosphate to 1,3-diphosphoglycerate. This is the oxidation reaction. First reaction of phase second. Now this was phase first. This was priming phase. 1,3-diphosphoglycerate gets converted to 3-phosphoglycerate. This converts to 2-phosphoglycerate. Lastly to phosphoenol pyruvate, you get now enol pyruvate, means finally you get pyruvate. Now this is irreversible reaction. First one was irreversible reaction, and this third one, that is fructose 6-phosphate to fructose 1,6, again irreversible reaction. Rest of the reactions are reversible reactions. Okay. Now this is the balance sheet. We started with glucose. This is the net reaction. Glucose is converted to 2 molecules of pyruvate. We started with 2 ADP, 2 NAD, and lastly we got 2 ATP molecules per molecule of glucose. So glucose gets oxidized to pyruvate. NAD, nicotinamide adenine diamine, this is a cofactor. This is in fact a coenzyme. NAD is coenzyme reduced to NADH, and ADP gets phosphorylated to form ATP. This happens over here. So starting with glucose under anaerobic conditions, we get 2 molecules of pyruvate, and 2, there is a gain of 2 ATP molecules. This is energetics or balance sheet. So again you can see from this chart, reaction number 1 we use ATP, reaction number 3 we use ATP, and there are other reactions where we get 4 ATPs, but initially we have spent 2 ATPs, one at first reaction, one at third reaction, so there is a gain of 2 ATPs under anaerobic conditions. So this is regulation of glycolysis. It is being regulated by 3 enzymes: hexokinase, phosphofructokinase, and pyruvate kinase. This is the enzyme involved in first reaction, phosphofructokinase involved in third reaction, and pyruvate kinase involved in the 10th or the last reaction. These very enzymes regulate glycolysis. Thank you.